Hi, welcome to my talk. The talk today is, can I trade you for a better phage defense system? Or the more boring title is, an experimental model system to explore the evolutionary consequences of CRISPR-Cas acquisition to a naive bacterial host. So why would you need a defense system against phage? Well, if you don't know what phage are, they're not very nice. They're the viruses that infect bacteria. They inject their genetic material into the cell, they replicate, they burst the cell. It's basically not a good time if you're a bacterial cell. So they have a variety of defense systems to try and defend themselves against the phage that are basically trying to murder them. And these defense systems are pretty fun. There's quite a lot of evidence now that these antiviral defense systems are getting frequently transferred between different populations of bacteria. So in terms of what we currently know about this, there is quite a lot of evidence phylogenetically and experimentally to show that there's quite a lot of horizontal gene transfer of these antiviral defense systems. You find them on islands that are frequently going between different populations. And we also have quite a lot of good evidence about the loss and degradation of these systems when you look across genomes. But what we have relatively little evidence for is if you are a bacterial cell who is sitting there and you just get yourself a new defense system, what happens next? What are the selective forces that you encounter, and how good is this defense system for your evolutionary trajectory? So that's what my PhD has basically been trying to address. Now, I'm not going to do every defense system. That's way too many. We're just focusing on my favorite defense system, CRISPR-Cas. So how does CRISPR-Cas work? Basically, viral DNA or any other mobile genetic element enters the cell, and the CRISPR-Cas acquisition machinery basically steals a little piece of that DNA, and it puts it into its own genome between tandem repeats. And this piece that it steals is called a spacer. And that spacer, when transcribed, is combined with nucleases, and that basically tells the nucleases where to go and what to cut up, which is a really great directed form of snippy things. And so the kind of overall aim of my PhD has been to model one of these horizontal gene transfer events. So we've taken the type 1F CRISPR-Cas system from Pseudomonas aeruginosa, a pathogen, and we've put it into Pseudomonas fluorescens, which is a soil bacteria that's been really well studied for its coevolutionary relationship with the lytic phage Phi2. So I wasn't going to sit around for my PhD and wait for a horizontal gene transfer to happen. I made one myself. So we basically ordered a synthetic version of the CRISPR-Cas system, and because we were ordering it, we were able to make it modular. So that means we put restriction sites around all the bits in it that you might want to change. And most importantly, we put a restriction site around the spacer, which basically means that at any point I can go in and change what it's targeting in the cell. And then we take this CRISPR-Cas system and we put it into the genome of the new host. So it's in there in one copy, and we don't have to have it under any selection to keep it there, which is really nice when you want to do evolutionary stuff down the line. So you're a bacterial cell. You've just been given a whole new CRISPR-Cas system by me. And the first question I want to know is, does this CRISPR-Cas system actually work? So to test that, we first were okay, let's target the CRISPR-Cas system against a plasmid. They're not fighting back as hard as a phage. So we gave it a spacer that basically matches the sequence of a plasmid, and then we ask, if it's got a CRISPR-Cas system targeting the plasmid, does this cause a decrease in the rates of conjugation into a new host? And what you can pleasingly see is that if you look at the wild type, it's got a conjugation efficiency of 6%. If you just put in all the machinery around the insertion, again, it's got a conjugation efficiency of 6%. If you give it the whole CRISPR-Cas system, but you don't give it a spacer that's exactly matching that plasmid, and again, it has a conjugation efficiency of around 6%. However, as soon as you give it the right spacer and a whole CRISPR-Cas system, suddenly you see this 2 or 3 tenfold drop in the conjugation efficiency, which is wonderful, great, this CRISPR-Cas system works. Maybe that's not that exciting because people use CRISPR-Cas for gene editing in heterologous hosts all the time. But what we're also interested in is, because we're using a native CRISPR-Cas system, can it get new spacers from incoming genetic material and basically do this almost Lamarckian-style evolution where it takes from its environments and evolves its own genome? And so again, we used a plasmid because they don't fight back as much. And we said, if we give you a spacer that doesn't quite match the plasmid, but nearly matches the plasmid, will you pick up any more spacers against the plasmid? And it does pick up a spacer against the plasmid. This will cause this regular 60 base pair increase in the size of the CRISPR array. So basically repeat, spacer, repeat, spacer, repeat. And you can see exactly that happen. So on this gel electrophoresis picture, you can see that some of them have gone up by like 1 level, which is a 60 base pair insertion, and some of them actually, they worked extra hard. They picked up 2 spaces, and they have this 120 base pair insertion. So this is fantastic. This whole thing works against plasmids. But that wasn't really who I was interested in. The real question is, can this CRISPR-Cas system actually target a phage in the new host? So this is a new phage it's never seen before, a new host that hasn't had a CRISPR before. So the way we follow this, if you're not so lucky as me to have set up 1,000,001 growth curves, we basically follow this over time in growth curves where at the beginning of the time there's not many cells, and as they proliferate in the liquid media, they increase the density of the media and the absorbance goes up over time. So if they're growing happily, you see this completely normal growth curve dynamic. And if bad things are happening in that liquid, such as you're being rampantly murdered by a phage, the line basically won't go up as much. So that's kind of what we're going to be looking at from now on. And we found this really interesting set of results that we weren't really expecting, that whether or not the CRISPR-Cas system in this new host, Pseudomonas fluorescens, actually worked, depended on the environment and it depended on the phage that it was targeting. So the first experiment we did was just your normal LB high nutrient conditions at optimal temperature. And you can see when there's no phage, all the lines are doing the same thing. And when you add phage, all the lines are doing the same thing, which is not what you wanted, because if you see this orange line, it has a CRISPR-Cas system and it's targeting the phage. And I didn't give it 1 spacer, I gave it 2 to be like doubly sure. And it's just getting like rampantly murdered by the phage, which was really not what I wanted to see. So then we decided to look into this a bit more. And we dropped the nutrient conditions because there were some other papers showing that that might favor the evolution of CRISPR-Cas. And what you can see is when you drop the nutrient conditions, obviously they don't grow as much in the presence of no phage. But suddenly when you add phage, this orange line that has the full CRISPR-Cas system and 2 spaces targeting this lytic phage Phi2, it's growing just so happily. Like, it's acting like there was no phage there at all. Like, the line is just completely happily doing this. So that was really excellent. And so then we were like, okay, well, what other environmental conditions cause this to happen? And there's this cool paper that came out from Padfield and et al. That showed that if you change the temperature for Pseudomonas fluorescens, it doesn't really have like a big effect on the growth of the Pseudomonas fluorescens. But as you get towards 30 degrees, you get this like complete drop-off in the efficiency of phage infections. So we were like, okay, let's play with this. If we run the same experiment, high nutrient conditions, but we just shift the temperature from 27 degrees to 30 degrees, and remember, this guy lives in the soil, like it's going to be experiencing a range of temperatures over its lifetime, we suddenly see this really powerful phenotype whereby you go from in the no phage condition growing happily, and then you add phage, and yet again, this orange line is growing completely happily. I realize the other lines are not very visible on this level of things, but trust me, there's other lines there for the controls. And so this was really interesting to see. And we were like, okay, well, is this an effect of the CRISPR, or is this something to do with this lytic phage Phi2? So we very kindly sent 2 other phage against Pseudomonas fluorescens, and we tested it against them. And as you can see, for both of these phage, if you do these in high nutrient conditions and at the optimal temperature, CRISPR seems to work completely efficiently against this phage, as though, it's able to grow in the presence of phage like nothing's happening to it at all. And this is really curious, because you imagine if you were doing a really massive screen to say, like, does CRISPR-Cas work against this phage? If you just did it in the, like, base conditions that we all normally use of, like, high nutrient conditions, optimum temperature, you would have said that CRISPR-Cas didn't work against this phage. But as soon as you sort of stray from those optimal conditions, suddenly you're seeing this really strong phenotype of CRISPR-Cas giving full protection against this phage. And similarly, if you tested it against a different phage, you see this really strong phenotype that CRISPR-Cas is giving this new host full protection against the phage. So, and there's another piece of data that I only just got, so I haven't added it into this slide, where you can see the same effect if you add in bacteriostatic antibiotics. So if you add just a sub-inhibitory concentration of bacteriostatic antibiotics, which obviously slow down all the machinery inside of the cell, you can also see this complete protective CRISPR-Cas phenotype, which implies our current hypothesis is sort of something to do with like maybe the time that CRISPR has inside the cell to like snip up that phage before the phage basically overwhelms it and takes over the population. But we're still trying to like unpick that at a molecular level. And so to kind of like conclude what this means, so when you give a shiny new CRISPR-Cas system to a new host that's never had a CRISPR-Cas system like this before, and you ask, okay, does it work? Yes, it targets a plasmid and it acquires space against a plasmid. If you ask, does it target a phage, the answer is it depends. So it depends environmentally between moving away from the optimal conditions, and it depends on the phage you challenge it with. And we also just had some data coming in. It's currently with the nanopore to get confirmed, but we've seen some limited acquisition of spacers against a phage, which is kind of, maybe not surprising that it's really, really tough for it to acquire new spacers against a phage because it's got to like take the spacer and then kill the phage before it dies. But we have, I think, seen a couple instances of it, nanopore confirming. And the final question I want to ask that I welcome to have anyone's input on how to best test is, does this provide a selective benefit to the host? Because if you look at those very strong phenotypes, you think that like, okay, great, like you're not having these huge genetic bottlenecks before you just mutate, say, the receptor that the phage binds to and like carry on to your next generation. However, other experiments in the original host, Pseudomonas aeruginosa, have shown that like the times when they want to use CRISPR-Cas seem to be sort of relatively limited. So the final part of my PhD in the last few months is hopefully going to be like taking all these conditions and say like, well, if you think of this like in the ecological system, like, are you actually benefit? Like, can you outcompete the wild type if I give you a new CRISPR-Cas system and challenge you with a host? And so I really hope the answer is yes, but we're still yet to see. And with that, thank you so much for listening. I would like to thank all the people who have supported me over my PhD. And you can't really see their names, but I promise they're there. Thanks. And if anyone has any questions, I do have a poster this evening that's an I1, so you're very welcome to come and chat to me.
Okay, it's 11:30, so hi everybody. Thank you all for being here, and thank you to the organizers for putting on this conference. My name is Helen. I'm a grad student in Paul Turner's lab over at Yale, and I'm excited to talk to you all about a fun project I've been working on lately. So phages we've already heard a little bit about, but what I'd like to sort of start off with is that phages do some things in nature and they do some things in the lab, and these things sometimes seem a little bit juxtaposed to each other. So for example, when we isolate phages from nature, these phages often have a high specificity to a certain bacterial host strain, and yet in the lab there's lots of experimental evolution studies that show that phages can readily expand their host range. So you find them often as specialists, but they could become generalists, and we're sort of asking, what's up with that? At the same time, phages, of course, depend on infection of their hosts in order to reproduce, but in the lab, many bacteria will evolve phage resistance quite readily. So as fast as overnight, it's very easy for bacteria to just lose their phage receptor a lot of the time and then become resistant. So the question that I like to sort of ask is, how do we reconcile these two things with each other? And one possible answer is this idea of community context. In other words, out in nature versus in the lab, you might have competitors, et cetera, that will affect how the evolution of susceptibility versus resistance is happening. And when we consider clinical applications such as phage therapy, we can see that the evolution and ecology of phages in communities actually becomes pretty relevant. So as an example, if you were to use a phage to treat a pathogen in a patient, and the pathogen then goes extinct or becomes rare, you might ask yourself, what does the phage then do? So maybe this very specific phage will also go extinct, but there's also a chance that the phage may begin infecting something like a commensal instead. And this, of course, could cause dysbiosis in the microbiome. So with this in mind, I set up an experiment in the lab using several different treatments, and they all have a different combination of a permissive host, so a bacteria that the phage can already infect readily, a novel host, so a host that the bacteria is, at least in its wild type form, is unable to infect, and then a phage. And for this talk, I'm going to focus mostly on the condition where we have both hosts and a phage together. So to talk to you briefly about who our players are, our permissive host is O157H7, which is a pathogen-derived strain of E. coli. The novel host is VW, which is a commensal E. coli and a common lab strain. And then the phage is this T2 family phage that uses OMP-C and LPS as its receptors, and importantly, these receptors have homologs in both hosts. And to just sort of help you remember, O157 is the O regional host, right? And then, of course, BW is the novel host, so hopefully you guys can remember that. So the first thing I'd like to show you is that when there's not phage there, O157 and BW actually play pretty well together. So over the course of this either 8 to 10 day coevolution, you can actually see that they maintain pretty stable ratios, and we're not really seeing big fluctuations or extinction of one bacteria versus the other. But when you add phage, this of course changes things. So the first thing that we see on the very first day that you might expect is that O157, which again is that original host, immediately takes a pretty deep downturn. So it's being killed off by the phage. In turn, BW is pretty happy and is growing up and doing quite well. So maybe you would think, okay, are we just going to see extinction of O157? But in fact, we don't. We see some persistence. And then what's sort of interesting is that around day 3, we actually see this flip, where O157 undergoes this big outgrowth, and in turn, BW actually suffers and becomes a lower population size. And then as we continue the experiment, we see this fluctuation between BW and O157 happen, again, where you see this sort of shift between one being abundant and then dropping, and then the other one growing up. And this is very repeatable. So these are 8 different biological replicates. And you can see that pretty much in every scenario, O157 dies off in the beginning, but then eventually grows back up, and along with that, BW starts to suffer. And then there's, again, another oscillation where O157 becomes rare and BW grows back up. Okay, so what exactly is driving the bacterial population dynamics that we're seeing? And in turn, what allows O157 to recover? And the sort of first, maybe obvious answer is that the phage begins infecting BW. So you have resistant O157, so it's phage resistant, right? And then the phage jumps to BW, that kills off BW, and that allows the resistant O157 to grow back up. So is that what we're seeing? In this graph, I'd like you to just really focus on the purple line, which is the phage isolated from each day of the experiment, trying to be plaqued on BW, which is, again, that novel host. And what you see is that by the end of the experiment, you do in fact start to see plaquing on BW, but if we remember from our previous population data, this sort of decrease in BW and outgrowth of O157 is happening around day 3, and you actually don't see plaquing of the phage on BW until later, if ever, in the experiment. So, is the phage infecting BW? Maybe, but plaquing evidence is limited. So with that in mind, we wanted to look a little bit more about general growth patterns of BW in the presence of this phage. So what I'm showing you here is some growth curves, which we're familiar with from the previous talk. And what we're looking at is wild-type BW grown in 1% supernatant from each day of passage. And pink here is the positive control, so this is just BW happily growing by itself. You can see that in early days of the experiment, the phage does not really affect growth of BW. But as we continue on, we begin to see 2 different growth signatures. One we can see in the gray and tan lines, and this is really what you'd consider to be sort of canonical of lytic phage infection. So we see that the phage is causing a decrease in optical density, so meaning a decrease in cellular density, and this is what you'd expect to see if the phage is lysing all of these cells. But we also see, we also see another effect where there's actually just a sort of depression in growth rate. So it doesn't look like BW is necessarily being lysed by the phage, but it is suffering a growth deficit. So is BW's growth impacted by the phage? The answer seems to be yes, even without the signatures of phage infection. So there's something that the phage is doing to BW aside from lysis, apparently. So if we want to ask what's going on exactly between BW and the phage, there's a couple things we can do. So for this graph, what I'm going to show you is if we put wild-type BW in with the phage supernatant from each day, and we measure the titer of the phage as plaqued on O157 before and after this 24-hour growth period, we can actually get an idea of is the phage growing, so to speak, or is the phage decreasing. So the first sort of obvious thing is that around day 3, we do in fact see that the phage is increasing by 2 log or more, just by growing with BW alone. So what this suggests is that even though we're not necessarily seeing plaquing, the phage is in fact reproducing and increasing its population size around day 3. Something that's sort of interesting, though, is that we also, around day 2, see this depression. So what we're maybe seeing is that the number of phage particles that you put in with BW actually decreases after this 24 hours. So this is suggesting that perhaps BW might be helping to relieve phage predation pressure on O157. And maybe then this is allowing O157 to regrow, as we see in the bacterial population data. So why might BW be taking phage out of the culture? And there's a possible clue here from qPCR, so I'm not going to get into the details of this, but basically what this graph is showing is the ratio of how much phage DNA we just have free in the supernatant versus how much phage DNA is contained within a viral capsid. So that would be a phage that was packaged correctly and has been made into a full viral particle. And the one takeaway from this graph is that, again, around day 3, you have a lot more free phage DNA present as compared to the phage DNA that's in a viral capsid. And this perhaps is indicative of abortive infection. Abortive infection is essentially the phage is able to bind and inject its genome into the bacteria, but the bacteria then undergoes lysis before the phage can successfully replicate. So in other words, this is a way that BW might actually be serving to take phage out of the population without actually becoming a reproducer of the phage. So to go over that quickly, what is driving these bacterial population dynamics and what allows O157 to recover, it seems to be a combination of things. One is productive infection of BW by the phage. In other words, there's more phage being made, and there is, in some cases, plaquing observed. The second is this abortive infection, and the third is also just this growth decrease that's seen in BW in the presence of the phage. And of course, this could be because of the abortive infection. So for the last couple minutes of the talk, I'm gonna speak a little bit about what seems to actually allow the phage to expand its host range. So the first thing we're gonna look at is a graph of phage plaquing on O157 that has this phage receptor, so OMP-C, or O157 that is a knockout for this phage receptor. And what I'd like to show you is that if you look at the very beginning of the experiment, so this is on your left-hand side, you can see that while there's plenty of phage that is able to plaque on wild-type O157, there's essentially no phage that is able to plaque on O157 that is not expressing OMP-C, which is what you'd expect, right, because OMP-C is the receptor. But as the experiment goes on, you see this arisal of a host-range mutant phage that is actually able to plaque on O157 without needing OMP-C. So you see some sort of change in what is required for the receptor for this phage. And then another possible clue is that if you look at then this other treatment, where we just have O157 in the phage, so BW is not in this treatment, we see that this evolution of OMP-C-independent infection happens also. And why this matters is because what we actually find is that host range expansion to BW can actually happen without BW ever being present in the culture. So this is a case of what we would call accidental virulence. So in other words, the phage has never seen BW, but as it co-evolves with O157, something is happening to the phage. Maybe as it undergoes this arms race dynamic or something with O157 in the phage, and that this just so happens to evolve the phage in such a way that it's now able to infect BW. So with that in mind, is the evolution of OMP-C-independent phage infection of O157 perhaps linked to the phage's new ability to infect BW? And I have some sort of just preliminary data to show you all, which is that, first of all, the phages isolated from the accidental virulence case that can plaque on BW do not need OMP-C. So these are OMP-C-independent phages, meaning that they're independent of BW's OMP-C. And yet, the phages that we isolate from the two-host and phage treatment do seem to need OMP-C. So I'm a little bit inconclusive so far about what exactly this means, but I would say that it seems that the way that you are co-evolving and the arms race dynamics that you're undergoing between a bacteria and a phage may in fact set the stage for how the phage, for the evolutionary trajectory of the phage, and this would then in turn perhaps affect things like accidental virulence. So of course there's open questions. What is the mechanism behind this accidental virulence? I'm still working on that. You know, are these host range mutant phages that we find in these two treatments the same, or are they fundamentally different? And the big question, I think, is what is happening genotypically to the population. So my hope is that through sequencing data, I will be able to really figure out what exactly is going on. And then of course, what is the phage using as its receptor? That remains to be seen. So some concluding thoughts. I hope that I've sort of shown you that phage bacteria dynamics are complicated, and that a lot of times it's not so simple as just being susceptible or resistant as being measured by plaquing. So there's a lot of ways that a phage can infect bacteria other than simply infecting it and lysing it, right? So when we consider population dynamics, when we consider community context, it becomes important to think about how phages are interacting with bacteria aside from simply lysing them. And with that, thank you all for listening, and I'd like to thank my lab, who's pictured here, and I'll take any questions. Thank you.
So considering that the OMP-C independent can infect
So I'm not sure yet. Does seem to still be OMP-C dependent phages, but the OMP-C independent phage seems to be at a pretty high frequency. What I'd like to do is see if 0157 is all mutant for OMP-C, right? And hopefully that will tell me, you know, is it because this whole population is swept to not having OMP-C or not?
This might be a naive question, but do you have any idea whether the phage interacts with the cryptic prophage genes that are present in the BW genome in any way?
Right, so I'm not sure about that. I did check to see that the phage I'm getting out is not prophage, so it is the lytic phage that I'm putting in. But yeah, I wouldn't be surprised if there's like an interaction between prophage or something that's then causing this sort of growth deficit. But that's definitely something I'll look into.
Maybe with the genomic data, it'd be really cool.
Exactly.
If you saw any recombination happening, but
Yeah, definitely. Yeah. Whoop. That was scary. Okay.
Oh, yes. Is that a sign? Thank you. It's a lot better. So when you were doing your original experiments that you were showing us, right, where you had the O and the BW,
Yes.
could you clarify, so were any of the phages that you pulled out from that experiment OMP-C independent, or is that a separate experiment that you did?
So I have run this experiment twice. And the first time it seemed that most of the phages I was pulling from that were OMP-C dependent. But when I ran the experiment again, I seemed to start to be getting this arisal of OMP-C independence. So there's something going on that seems to be a little complicated. It doesn't seem to be quite as black and white, and I'm hoping that the sequencing data will maybe tell us a little bit more about that.
So the next speaker has withdrawn, so we can do extended questions from the first two talks.
Yeah.
I have a feeling you already know this. I just couldn't see that slide.
Oh, no.
It looked like you did spot tests.
Yeah. Did that.
About couldn't you use? Yeah, they're lovely.
Could this not show up? Sorry.
Yeah, couldn't you use those to figure out about the word of infection, whether that's really happening?
Yeah, I have to read more about the abortive infection because I do see sort of like these halo like plaques, you know, where it's not really a plaque, it's just a clearing. And I do suspect that could be abortive infection. I also kind of wonder, so all these spots are on the ancestor, right? And I do wonder if maybe some of the morphology I'm getting in the plaques is actually just because the wild type host isn't really there anymore, and I would need to pull a host from somewhere in the middle of the experiment.
I just think even if they can't make a plaque, if
Yeah.
they can interfere with the host growth,
Right.
show up at this assay.
Right.
Not in a plaque.
Yeah, that's something I
Yeah,
definitely.
I'd love to chat more with you.
Yeah, definitely. Yeah, thank you. But that's a really good point. I'll definitely look into it. Yeah.
How are you hoping to look for the potential like new receptor then, or do you think it might be a mechanism that's almost like a little bit receptor independent?
Yeah, so I have a few thoughts. So one thought is that there is a different outer membrane protein that is exactly the same in both of these hosts. So I have a suspicion that if the phage starts to use that protein as its receptor, then of course it would be accidentally virulent because it's the same protein. I'm not super sure if that's actually what's happening. So I've done some sort of like looking at different knockouts, right, and trying to figure out what's going on. I think what I'll probably have to do is really just sequence some resistant isolates and try to get an idea of what's being mutated, and maybe that will be clear. Maybe it won't, but yeah. Other questions? Okay, awesome. Thank you.